Hello, everyone. My name is Tegan Clary, and I head up the marketing team at Unchained Labs. I'll be your moderator today, and thanks for joining us. We will have a Q&A session at the end of the presentation. So to ask questions, all you have to do is click on the Q&A button in the Zoom navigation bar, type in your question and send it on in. We'll get to as many of them as we can today. I would ask, please don't submit it anonymously. So if we're able to, we'll be able to reach out to you and follow up if we're not able to get to your question, or there might be a more in detail answer. Again, please don't be shy, submit questions. Rick will love to answer them. And now I would like to introduce Rick Sidler. How are you today, Rick?
Great, Tegan. Thanks. Nice to be on a webinar with you again.
Yeah, same here. Always fun. Rick is our Director of Field Automation Scientists here at Unchained Labs. Rick will be taking us through how Junior can help you deal with finicky biologics, getting them into the right formulation, specifically by automating activities like visible particle counting, pH measurements, and viscosity measurements, as well as some other really interesting and cool techniques that we've been able to add onto our Junior platforms. And Rick will talk a little bit about our Big Kahuna platforms as well. So Rick, I think I'll hand it over to you. I'll come back and we'll do some questions at the end. So take it away.
Sounds great. Thanks, Tegan. Hopefully my slides are up now. And
they are, and they look good, Rick. You're ready to go.
Awesome. Thanks, Tegan. Welcome, everyone. Glad you could join us today. Biologics are finicky. Each one is different, but they're certainly worth the effort because they're critical to treating and managing human illness. Oftentimes additives or excipients such as salts, amino acids, sugars, surfactants are common and need to be identified for each specific formulation. Some can be frozen, some can't, some can be lyophilized, others no. But typically, they need to be kept in aqueous solution with highly specific storage conditions. Viral vectors are finicky too. They're hard to make, they're challenging to purify, and the results are typically very expensive to produce. So we need to treat these with a special care and to search for the best formulations and storage conditions to optimize their longevity and potency. Most are encumbered with challenging storage conditions. This one must be stored at -65 degrees. That's not actually too bad. But others are a lot more daunting requirements. Here, this particular one has a tight temperature range. It can't be refrozen. You have to use within 14 days. And to top it off, it can't be shaken. With all of these challenging conditions, analyses of these biologics and viral vectors are critical to ensuring their potency, efficacy, and stability. Well, pH has been called the most critical attribute for formulation of proteins. And so pH typically is controlled and tested for everything. Here we see the effect of pH on solubility of a monoclonal antibody. Variations in the excipients show different solubility behaviors as a function of pH. But there are many other parameters and analyses to consider also. Visible particle detection for materials greater than 80 microns, subvisible particle counting, and aggregation are also key assays. Typically, you need to measure for these samples at different stressing conditions, such as a variety of temperatures, 5C, 20C, 40C, and at various time points out to and beyond, oftentimes 12 months. Shaking also at various temperatures is important, as are the effects of freeze-thaw cycles. This can easily lead to hundreds or thousands of analyses that need to be performed and determined for the best formulation and storage conditions. Some of these assays are relatively easy to perform. Others aren't so pleasant, either because they have challenging sample preparation or just tedious, slow, busy work. Fortunately, automation can help with many of these tasks. Here, Amgen is using full-scale automation to perform buffer exchange, measure pH and viscosity on a large integrated platform. Look at the significant reduction in key metrics, such as the hands-on time for buffer exchange and pH measurement, both reduced by over 70%. And a 90% reduction in hands-on time for viscosity measurements, leading to the screening of more formulations much more efficiently. You can do that also. If your throughput requirements are high and your bottlenecks are big, this might be the right solution for you. But you can also use the smaller Junior platform to automate things that matter to you or are bottlenecks for your specific workflows. These smaller platforms are typically less expensive, take less room in your lab, and are more approachable for occasional users, allowing you to automate your testing and expand your formulation space. They can be configured specific to your needs to help with formulation and characterization of myriad biologic and gene therapy materials. Here's an example of a junior deck for analyzing biologic formulations. This system has a viscosity station, visual inspection, and the ability to store many plates of samples for processing. Let's take a look at some of the options you have for automated assays. For pH measurement, we have a 4-channel glass micro pH probes that can measure in everything from pharma vials down to PCR plates. These probes have a wide pH range with high resolution, and importantly, a fully automated calibration, measurement, washing, and storage cycle to free up your time. Here you're seeing that washing cycle in action. When we look at the data, these are real samples of IgGs in a range of different pH buffers. The probe spacing measures every 4th sample before shifting over one well for the next set. In this particular set of data, all are measured 30 times. Look at the accuracy relative to the manual method that you see. Can you even imagine the amount of work involved to manually measure and record 30 replicates of 16 different buffer samples? That's 480 individual pH measurements. With recording the results and washing the probe, if it took you a minute per sample, that's 8 hours worth of manual work. Certainly not how I want to spend my day. Probably not your idea of fun either. With automation, you just set it up, then go do something more scientifically rewarding or challenging and let the robot perform the drudgery. Visible inspections another measurement that's mind-numbingly boring. It's not hard to do, but you can't do it for too long at a stretch. Automation is great for this task. We have an image-based visual inspection station that performs particle detection and counting, color analysis, and turbidity measurement. These all happen from image analysis, and the images are stored so you can always go back and look at them later if there are questions or issues that arise in your formulations. So how do these work? Samples are spun in their closed containers. This is a non-invasive technique, and that gets the liquid and any particles moving around. Then we stop the spinning and capture a series of images while the liquid continues to move. We subtract pairwise sets of the images. Anything stationary goes black, while movement between images is recorded as a lighter area, which is detected and counted. For color measurement, we also rely on imaging. We measure and store a set of color standards, here shown in the top row of the BY color set. Then we image your samples and we compare the color of those to the standards to determine the color match and the relevance score for your materials. Turbidity is also based on image analysis, but in this case, we're looking at the luminance channel for the color space. This value goes up as the turbidity scatters light. You create a calibration curve from turbidity standards, and then you compare your sample readings to that curve to get NTU values. You can even create two-step or two-section calibration curves, so you can get better accuracy at the low NTU values and at the high NTU values. Viscosity is also a critical parameter for many injectables. We have a high throughput capillary viscometer, which is completely automated, everything from sample injection, washing, and temperature control. And key, it only takes about 2 to 3 minutes to measure each sample and requires only about 100 microliters a sample. For real protein formulations, here is some data for automated viscosity measurements, 96 samples in 7 hours. That means the ability to measure over 300 samples in less than a 24-hour working day, requiring only 100 microliters a sample. It's really pretty amazing when you consider it only takes a few minutes of hands-on time to set this up. Well, mostly, we've been exploring some of the integrated analytic techniques for evaluating your formulations, but we know there's so much more to the overall process in your labs. Early in the webinar, we discussed storage requirements for some biologic and gene therapy materials. A critical test for this is the freeze-thaw cycling. While many monoclonal antibodies might be okay with being frozen, this isn't necessarily the case for other gene therapy products like LNPs or AAVs. Each one's different depending on the material and its formulation. The rupture of AAVs during the freezing or the thawing process is often a problem and may require additives such as cryoprotectants to mitigate. Ideally, during the testing, you need to pull samples for evaluation after each cycle. In practice, these can often come at very inconvenient times, like in the middle of the night or on a weekend. Automated freeze-thaw testing can be the answer. We've automated this with a freeze-thaw block that can be added to your Junior or your Big Kahuna. After formulation, the robot can place the samples in the freeze-thaw unit, add a cover which sort of protects the samples purged with dry air from frost and condensation, and then can cycle the temperature. You get to control the number of cycles and the parameters such as the temperature and the time. After freeze-thaw, you can analyze the sample by non-destructive techniques such as the visible analysis we discussed earlier. Or if you want, you can have the robot open each container after each cycle and pull sample for analysis such as DLS or analysis of the free DNA, etc. You can even go further with integration of the DLS measurement right on the robot deck. Here we have a Stunner DLS instrument integrated on the Junior platform. You can use the system to deposit the samples into the analysis plate and move it into the instrument for analysis. Everything is automated. When we think about your overall workflow, it usually comprises a variety of different steps. We often think of these in groups where you first prepare a bunch of materials that you want to study. You then perform some sort of processing steps on those, followed by analysis of those samples. Mostly today, we've talked about the techniques of pH measurement, visual inspection, and viscosity, along with a brief discussion of freeze-thaw, but we really have you covered on all of these different techniques and many more. Whatever your challenges, whether you only need to address a few bottlenecks with a Junior, or you want a larger, more fully integrated workflow, Unchained Labs has you covered. I hope your interest has been piqued during the presentation, and it's time to develop your formulations that really pamper your biologics. At this time, I'd be happy to turn this back over to Tegan for some questions from the audience. I'd like to thank you for your time and for your attention.
That's great, Rick. Thanks for taking us through that. Just to recap, I think, on Junior systems and even into our Big Kahuna systems, you can automate pH, visual inspection, viscosity measurements, and you can do that all on a single robotics platform. And also, we do have a lot of really interesting techniques and tools that we've worked with customers to develop, such as the freeze-thaw, or some of those integrations like DLS or other tools like from Unchained Labs or from other companies. So I hope everybody doesn't leave this thinking that, hey, there's just these sets of things. It's pretty much a wide open field when you come talk to us about what you're trying to do. And if we can do it, we're willing to talk to you about it. So, and Rick is one of the key people you'd be working with, or his team, to actually put together that vision. So Rick, thanks for taking us through that. Let's go ahead and hit on some of the questions. Okay, you ready for those?
I am ready. Bring them on.
Okay, first one, Rick, is I already have multiple liquid handlers in the lab. Can a Junior robot with pH and freeze-thaw be integrated with other systems in my lab?
Yep, absolutely. There are a number of ways you can do that. You can just move samples to the system, do the analysis, and move them back. We also have an API, and you can integrate with plate moving robot arms to transfer samples there, kick off the analysis or the runs for your pH measurements or your freeze-thaw studies, and then move samples back to the deck of your other robotic platforms as well.
Great. Another one, how do I view the data from the various analyses like the pH and viscosity? Where's that data go, and how do people look at it?
Yeah, the data all goes into a database and it's stored. And so you can actually go back and view it, whether it's image analysis from the visual inspection or data from pH measurements, as the question was for. We have a variety of tools that allow you to go in and look at that. You can generate some sort of standard reports where you can get a PDF output of the vial or container position along with the pH values. We also can store those in an Oracle database, which you can access and generate reports using either PolyView or LIA analysis, which are our sort of flagship tools for data viewing.
Okay, another one here, Rick. How long does it take to set up samples to run the visual inspection station on our systems?
It's pretty quick. You just enter the samples into a library design, so we know what the plate format is and where the samples are. It takes just a minute or two to create that. And then you would start up the robot software, Automation Studio, to do the run. With Automation Studio 10, that only takes a minute or two to get started. So definitely just in the range of a few minutes and you can be up and measuring.
Okay. Rick, one more here. I think somebody's asking who else out there besides Amgen is using systems like these for biologics work, and then are there any companies we're able to mention that are doing gene therapy workflows? We can also take that offline if it's not appropriate, but I think companies that have published, there's quite a few. So which ones have you worked with, Rick, that we can talk about, or that are references for other people?
Yeah, we do have systems that pretty much most of the large and mid-sized pharma companies and a lot of the biologics gene therapy companies as well. Some of those that have published, BMS has been a big user of our systems and big publisher. They've also presented a number of user group meetings and other sort of public forums there. It's probably better for some of the other companies if there are specific ones we can reach out to direct customers with. Yeah,
so anybody that does have that question or would like to talk with us about a specific workflow or specific challenges, our customers are often very willing to talk to other people that are thinking about making investments in solutions like this. So just reach out to us. We'd be happy to talk to you first about what you're trying to accomplish, and then potentially take that to someone that has a similar solution so you can connect with them. So don't be shy, and you're always welcome to ask us for those connections. Okay. All right, Rick. Well, I think that wraps up the questions. Thanks for answering all those. I want to thank everybody for joining us today. And if you would like to have a deeper conversation with us about your problems, your workflows, your bottlenecks, you can always reach out to us at info@unchainedlabs.com, or you can head over to our website at www.unchainedlabs.com. Take a look at the Junior and Big Kahuna products. They're under our product section. And the other place that I always want to plug is go to the Learn section, and you can click on the checkboxes for Big Kahuna and Junior, and you can see a lot of presentations like this on different topics, and some of our customers that have presented in the past. So you can take a look at that content. So again, thank you for joining us for this virtual seminar. I hope you have a great rest of your day, and hopefully we'll see you again soon. Thank you. And thanks again, Rick. Great job.
Thanks, Tegan.
All right. Bye-bye.